Today, our group will be presenting an RNA sequencing analysis of the bilin signaling pathway in HMOX1 and wild-type C. Reinhardti cells. So, why do we care about bilin? According to Dunmu's study, bilin is a key signaling molecule in the regulation of photosynthesis. In most plants, bilin-containing phytochrome responds to red light stimulus and coordinates genes involved in photosynthesis, giving photoautotrophs an adaptive advantage during daylight transitions. Chlamydomonas reinhardti lacks phytochromes, but bilin biosynthesis in this species still exists, and is done by heme oxygenase and bilin reductase. Therefore, we would like to confirm whether or not bilin plays a role in photosynthetic gene regulation. Based on the rationale that HMOX1 genes code for functional enzymes that lead to the biosynthesis of bilin, we hypothesized that if bilin-dependent signaling exists in C. reinhardti, HMOX1 mutant cells, when exposed to light, will lack the photosynthetic response seen in wild-type cells. This will be reflected through differential expression of photosynthetic genes. In summation, we aim to find differentially expressed genes between the mutant and the wild-type cells.
In our study design, our main study question is, does bilin-mediated light-dependent signaling exist in C. reinhardti? The two main treatments used were HMOX1 mutant and wild-type cells with 2 replicates each. To determine the number of differentially expressed genes, we compare light-dependent responses in mutant and wild-type cells by analyzing mRNA expression and using the R program. Two limitations of the design include lack of biological replicates, as sampling effort can be increased, and the difference in genetic background between wild-type and mutant is relatively unknown. Alternatively, we could use HMOX2 mutant cells or measure gene expression using ribosomal profiling approach, capturing changes in photosynthetic protein abundance. Compared to the mRNA measurement, the ribosomal profiling approach is more accurate in reflecting the result, but it can be difficult to identify all the photosynthetic protein.
We use the correlation heat map and the principal component analysis to assess the quality of the samples. For the heat map, most biological replicates are clustered together by the dendrogram, which suggests that the sample labeling should be correct. One exception is that the homox treated with bilin is clustered together with the untreated homox samples in the dark condition. However, it makes sense because bilin treatment is not expected to cause any effect in a dark environment, and thus, the gene expression in mutants with or without bilin treatment should be very similar. For PCA, again, most biological replicates are clustered together. Moreover, the PCs also correspond to the variables in our study design. PC1 captured the variance between the mixotrophic and autotrophic cell types. PC2 captured the variance between the dark and light condition. PC3 captured the variance between the wild-type and mutant strain. And PC4 captured the variance between the bilin and non-bilin treatment for each cell type. We made 3 contrasts to address our study question, and all contrasts are for the dark to light transition. Contrast 2 and 3 aims to capture the differentially expressed gene in wild-type and mutant. Contrast 1 aims to identify those genes that respond to light differently between wild-type and mutant cells. As we described before, PC3 should capture the variance for this contrast.
By comparing the two controls, contrast 2 and contrast 3, we can observe that they both exhibit similar patterns in the number of up and down-regulated genes. However, contrast 2, or the wild-type cells, has slightly more regulated genes. According to the top plot, there is still a significant number of genes that respond to light differently between wild-type and mutants, which indicates that the mutation does affect the light-dependent gene expression. According to the first contrast red circle of the Venn diagram, there are a total of 1,340 genes that respond to light differently between mutants and wild-type cells. More specifically, when experiencing dark to light transition, 1,033 of them are more upregulated in mutants compared with wild-type, and 307 of them are more downregulated in mutants compared with wild-type. Notably, as you can see in the green circle, there are 389 genes that only differentially express in mutant cell during light to dark shift. These genes might be responsible for the survival of cell in the absence of photosynthetic activity, or they could be abnormally regulated due to HMOX1 deficiency. Our hypothesis is supported due to the fact that 1,340 genes were differentially expressed between HMOX1 mutant and wild-type cells in response to changes in light conditions, assuming that the differentially regulated genes are photosynthesis-related. These results confirm that bilin plays a significant role in the regulation of light-responsive genes, particularly during the transition from dark to light. These genes are good candidates of photosynthetic genes, which can be further investigated in future studies.